Welcome back for more AP Bio. This week, or at least this particular lecture, we're going to talk about population genetics, which is going to be the fusion of genetics and evolution into one organism. The College Board really likes this one, so it's going to show up on the AP test. So from once upon a time ago, we dealt with Mendel's laws, and Mendel came up with the data that derived three laws. The first one is the only way to explain the patterns that he saw was for each trait, it has two different versions or two alleles that segregate or separate from each other. Law 1. Second one is one of those alleles can cover up another allele, the law of dominance. The third one is traits are inherited differently, meaning a trait for the color of flowers has nothing to do with the trait that deals with the color of peas. They're totally separate from each other, hence there's independent assortment. So we had three rules that allowed us to deal with genetics problems, but we also had Hardy-Weinberg equilibrium, which basically established there's no evolution. How'd we do that? By saying 5 conditions. So we had large populations, there's no net mutations, we have no natural selection, we have random mating, and we have no net migration. What we can do is take those two and merge them together, and when we do that, what we start noticing is we can create a hypothetical situation called a gene pool. And a gene pool is for any population, for any given trait, it's all of the alleles that are in that particular population. So if we have 100 individuals, it's the 100 individuals worth of alleles for how straight hair is, or the 100 people's worth of alleles for whether or not you have a hitchhiker's thumb or not. You throw all those in, and what we can do is we can calculate out frequency. So how often do you see this allele or that allele? What I can do is take that and put them into some equations. These are referred to as the Hardy-Weinberg equations, and they apply, especially that last one, if we're in an equilibrium situation. The first one, P plus Q equals 1, that's interpreted as the gene pool, and that would be the dominant allele frequency, P, and the recessive allele frequency, Q, equals 100%. The bottom equation, P squared plus 2PQ plus Q squared equals 1, that one there deals with individuals. So what percentage of the population is homozygous dominant, P squared? What percentage of the population is homozygous recessive, Q squared? What percentage of the population is heterozygous? That's 2PQ. What I can do is take these bits of information and I can turn it into math problems that actually apply to real life. There are two different versions of these Hardy-Weinberg equilibrium problems. The first one is straightforward, and it's just you're finding out percentages or you're finding out numbers. So what you typically do is you're going to solve for Q squared first, so find how often the recessive individual shows up. Use that to solve for Q. You're then going to solve for P. Once I have P and Q, I can solve for anything else I want. I could find P squared, I could find 2PQ, I could find numbers who are P squared or 2PQ. I can do all sorts of stuff like that. The second problem is, am I actually in equilibrium? For that type of question, are we in equilibrium or not, we have to take a slightly different approach. The first question is, solve for P and solve for Q. You're going to do that using the raw data that you're given. What you could then do is actually you take frequencies, so I have P and Q, I can now find P squared, 2PQ, Q squared. I can either take those percentages as percentages, not as decimals, and I can compare that to what I actually see for P squared, 2PQ, and Q squared, or I can take the numbers of individuals and convert my P squared, 2PQ, and Q squared into actual individuals. We talked about this in class. I'm not going to do a math problem here, even though it's going to say I have one. I'm actually not going to do it. And then we do a chi-square. Seems easy enough. This is the example that I gave. This is actually a type 2 problem where I give you data for individuals. What I notice if I read everything is the A is going to be dominant, so it's going to be P. G turns out to be recessive, which is Q. I can count the number of A's I have, divide by the total numbers, which is 42 for this A, 42 for this A, 24 for this A, 24 for this G, 21 for this G, 21 for this G. Turns out to be 174. I take up all my A's, divide by 174, that gets me P. I could take the remainder, 66, divide by the 174, that'll get me Q. I can now get p squared, 2pq, q squared. Total number of individuals is 87, so it'd be 87 times the percent of p squared, 87 times 2pq, 87 times q squared. I can now do a chi-square. The point is we can now start applying math. So I can actually calculate things that we call fitness, which is this omega symbol down here, that W. So how good is this trait or this combination of traits? How well does this let you survive? We can calculate that out. We can calculate selective coefficients, which are S's. We can deal with heterozygote advantages, which is an H. We could find mutation rates, which is mu. We could find migration rates, which is an M. We can find out all sorts of things. And what I can do is use these numbers to predict the future and see, is that what happened? We have an ability to mathematically model what we see happening and see what it does in the future. Which means we now have a new way to define evolution. And instead of talking about how the bones have changed or how footprints have changed or how locations have changed, instead what we can now define evolution as is changes in allele frequencies over time. Because that, we just argue over the sources of the data, but the number is the number. And now we can just track that out. If we're dealing with natural selection, the trend is going to be obvious. There's going to be an obvious pattern to what's going on. Because a selection is a conscious choice when we use the word selection. I am choosing this over that. So what I should see is an obvious pattern, an obvious direction that we're going. And that results in evolution. Natural selection, we should see an obvious pattern. Genetic drift, on the other hand, we won't see such an obvious pattern. And what we get is what we call a stochastic type of graph. Stochastic, a random, a variable, I don't know what's going to happen next graph. And as you look at these three, the major difference between the first one, second one, and third one is the number of individuals. They both go through 50 generations. Both of them have the exact same y-axis in terms of allele frequencies. The difference is, as we go to a population of 2,000, there's a pattern. There's an obvious flatline pattern that exists that was not there in the first option. And that's because genetic drift is the randomness of reproduction. And the randomness trumps the overall pattern, which we see as the population gets bigger. But we can track this using allele frequencies. So all of this has led into something we refer to as the modern synthesis of biology. This is going to be the integration of genetics and evolution. And when those two are combined, what we end up getting is an ability to view anything in the world of biology in the terms of evolution and in terms of genetics. So what we're going to be doing for now on is we're going to be combining evolution and genetics because the two are quite explicably linked together.